Welcome back, everyone. So what we're going to look at here is this idea of how we can regulate our GFR. So in the previous part, we talked a little bit about some of the pressures, right, that can influence or that promote this process of filtration. We talked about net filtration pressure, and then we talked about glomerular filtration rate, or GFR, right? And again, so remember, GFR. This is just the um, amount of uh, blood that's being filtered, the amount of filtrate that we are forming over some given unit of time, right? You can think of it in milliliters per minute. How many liters per day do we make, right? That 125 milliliters a minute, 180 liters a day, something like that, right? So there's two main kind of mechanisms or processes by which we regulate our GFR, right? And these are called the intrinsic and the extrinsic mechanisms, or these controls, right? So meaning the intrinsic meaning is it something that the kidney itself is able to do to regulate GFR, either cause it to increase if necessary or decrease in order to maintain that consistent level, or is it something that is outside of the kidney? So think like maybe nervous system, think hormones that influence the kidney's GFR, right? So we'll look first at some intrinsic mechanisms, and then we will look a little at some extrinsic mechanisms, right? So intrinsic, again, think of the word intrinsic, think internal to the kidney. So something that the kidney itself is able to do to maintain a relatively consistent, constant GFR, right? So the first one of these is called the myogenic mechanism. Okay, myogenic. Think of what that prefix myo means. And then the second one is called the tubuloglomerular feedback mechanism, right? So we'll look first at our myogenic response, right? So the myogenic response, what I want you to think of is think muscle, right? Think muscle. So we're having something to do here with muscle. What kind of muscle? Probably smooth muscle, right? We're not really talking about skeletal muscle here. We're not talking about the heart, so it's not cardiac muscle. So this is smooth muscle, right? So two slides here. This is showing what happens if you have a decreased blood pressure. This is showing what happens if you have an increased blood pressure, and both of those you can see on this image here. So you can review these slides. Again, I say see image on upcoming slide. You can review these two slides here, but you could also look at this graphic, right? And so this is what I'm going to focus on. So what you see here is in the middle, this middle image, you see a quote unquote kind of normal blood pressure, right? And I should mention that our GFR stays relatively constant. It stays pretty steady so long as we have a mean arterial pressure, right? The MAP that we've talked about before, that mean arterial pressure of somewhere between 80 and 180 millimeters of mercury, right? So as long as we have the MAP between 80 and 180 millimeters of mercury, our GFR is going to stay pretty constant, right? It's going to stay relatively steady, right? And so whenever we have changes in blood pressure, such that our MAP changes in one of those two extremes, right? It's below 80 or it's above 180, then we can begin to have these different intrinsic mechanisms, right? So again, this first one, myogenic. So the idea here with the myogenic mechanism is that you have changes in the arterial smooth muscle as a result of different amounts of blood flowing into it, right? So specifically what we're talking about here is the afferent arteriole, right? So remember, if you think of your kidney anatomy, right, you've got the afferent arteriole, you have the glomerulus, and then you have the efferent arteriole. So the afferent arteriole, it is, under normal conditions, it is larger, it's wider, it has a bigger lumen than the efferent, right? And you can see that here in this middle image. So that means that more blood comes in, it gets kind of bunched up in these capillaries, and then the smaller efferent arteriole means that the pressure inside the glomerulus is going to increase, right? And that's that glomerular hydrostatic pressure we talked about earlier. That's going to promote filtration, right? So that's under normal conditions, right? So if we have a decrease in systemic blood pressure, right? So think systemic blood pressure in general, right? So think your MAP overall is say below 80, it's below 60, something like that. What you're going to end up have happening is because there's less blood flow in the body in general, that also means that there's going to be less blood flow to the kidney. So there is going to be less stretch of that afferent arteriole, right? Now, one thing to note about smooth muscle is that it constricts and dilates in response to being stretched or not stretched, right? So in this case, we're not sending a lot of blood into the kidney because we have an overall low blood pressure in the body, right? A low MAP. And so there's not a lot of stretch of that afferent arteriole. So the smooth muscle is going to dilate, right? So what you have happen is that afferent arteriole dilates. Well, if you remember the afferent arteriole, this is what's bringing blood into the kidney. So if the afferent arteriole dilates, what does that mean for the amount of blood coming into the kidney or into the glomerulus in particular? It's going to increase, right? So if now we've increased the amount of blood flowing into the glomerulus, we're going to influence and increase this glomerular, blood hydrostatic pressure, and that's going to help promote a positive net filtration pressure and thus increase GFR, right? So again, widened arterial lumen allows more blood into the glomerulus to offset a decrease in systemic blood pressure, right? And again, we're thinking here on the extremes, right? A really, really low blood pressure or really, really high blood pressure in our next example, right? So if you know what happens here when you have a decreased blood pressure, then you already know what happens when you have an increase, because it's exactly the same except that it's totally opposite, right? So in this case, if we had a decreased systemic blood pressure and we vasodilated, right, our afferent arteriole got larger, then when we have an increase in our MAP, our increased systemic blood pressure, we're going to have the exact opposite. So now we're going to have vasoconstriction, right? Why? Because as more blood, right, as we have a higher systemic blood pressure, we're going to have more blood flowing into the kidney, right, into the afferent arteriole. That additional blood flow is going to cause stretching of the afferent arteriole. And as I mentioned a second ago, stretching of smooth muscle is a stimulus for the smooth muscle to constrict, right? So we send more blood into the kidney, more blood stretching the afferent arteriole. That's going to cause the afferent arteriole to constrict, and a more constricted, narrow afferent arteriole allows less blood into the glomerulus, right? So that means that your blood glomerular hydrostatic pressure is going to drop, which is going to decrease your net filtration pressure, which is going to decrease your glomerular filtration rate, or GFR, right? Again, keeping with trying to maintain a quote unquote normal GFR, right? So again, a normal GFR being 90 to 120, 125 milliliters per minute, that 180 liters a day, right? So this is the myogenic mechanism, right? So think myogenic muscle, smooth muscle in our afferent arteriole, right? The second one, which is kind of a backup to the myogenic, is called the tubuloglomerular feedback mechanism, right? This one is slightly different. It acts in a totally different way, but it's acting to ultimately result in the same thing, right? So again, the idea here is we're trying to keep our GFR constant in responses to changes in kind of systemic blood pressure, right? So what's happening here with our tubular glomerular feedback mechanism is when you have an increased blood pressure, right? So increased systemic blood pressure, we're going to initially there's going to be more blood flowing into the kidney, so you're going to get more filtration, and you're going to have more solutes, more things in the tubular fluid than you would otherwise, right? So you've got this excess amount of fluid in the capsular space. There's excess solutes as well because all of it got forced out under this increased pressure. And so as this uh, tubular fluid is flowing through the nephron, right? It goes through your proximal tubule, proximal convoluted tubule, descending loop of Henle, and then it goes up the ascending loop of Henle. And part of that ascending loop of Henle travels right next to the afferent arteriole, or kind of in between the afferent and efferent arterioles, right? So I'm going to show you a picture here, and you can see this, right? So if you look on this right image, here's your proximal tubule, here's your descending loop of Henle, here's your ascending loop of Henle, and again, it's passing between your afferent and efferent arterioles. Here you can see the same thing, but more of kind of a cross section. So here's an afferent arteriole bringing blood in, here's your glomerulus, Here's your efferent. Here's part of your proximal tubule. It would go, so think coming over here, think descending, and then ascending loop of Henle, and this is a cut section of it, right? Part of that ascending loop of Henle is a region called the macula densa, right? So this term here, macula densa, right? So think of this, and you can see the word here, densa. It is this region of the afferent arteriole that kind of butts up next to the afferent and efferent arterioles that is very dense in cells, right? So think densa, dense, right? So you have this row of cells, and you can actually typically see this histologically as well. If you're looking around at the tissue, the histology of the kidney, you can sometimes see this macula densa pretty well. Think of this macula densa as uh, containing receptors that monitor the composition of the tubular fluid within this ascending loop of Henle, right? And so again, if you look back at this slide, the amount of the solute, sodium chloride, is increased. So it's going to be detected by those macula densa cells, right? So what ends up happening is the um, the the ascending loop of Henle, right? It's detecting this increased amount of solutes, and so what you end up getting is for that afferent arteriole that it runs right next to, right? So you're going to remember it's running right next to this afferent arteriole. You're going to end up with constriction of that afferent arteriole in response to the increased, solute concentration of the ascending loop of Henle. So if you have constriction of the afferent arteriole, and you've got these smooth muscle cells, these JG cells are called juxtaglomerular cells that are part of this whole region here called the juxtaglomerular apparatus because it's next to the glomerulus. You'll end up with this constriction of this afferent arteriole that's going to decrease the amount of blood flowing into it, which is going to decrease net filtration pressure, decrease GFR, right? And so you can kind of read through this like summary here, right? So again, we've got the detection of the filtrate by the macula densa cells. The afferent arteriole constricts, resistance in afferent arteriole increases, hydrostatic pressure in the glomerulus decreases, right? That's like glomerular or blood hydrostatic pressure. Your GFR is going to decrease, and now you've reversed the initial issue, right? Which in that case was increased solute concentration secondary to an increase in blood pressure, right? You're forcing more fluid out of the glomerulus into the capsular space. There's less time for it to be reabsorbed, so more of it ends up in your tubular fluid, right, in the nephron. Okay. So again, you can see these are kind of showing similar things, talking about our kidney's ability to auto-regulate itself, and then what happens if you have a low blood pressure or a really high blood pressure. Again, note the ranges there, so 80 to 180 millimeters of mercury. So you can look at this. You can also look at a graph like this. I find this to be kind of a helpful way to visualize this, right? So again, everything in here in this kind of blue zone, 80 to 180, we're going to maintain a normal amount of urine production because we're going to maintain a normal, quote unquote, normal GFR, right? Because of that auto-regulation that we just talked about. So myogenic mechanism, tubuloglomerular feedback mechanism. When you get into either extreme, right? You get into either the really, really high maps or the really, really low maps. Now you're going to see more significant changes or more visible changes in your GFR that's going to result in significant changes in urine production as well. Right. And so these two things, again, these are your intrinsic mechanisms, myogenic and tubuloglomerular feedback. What we're going to do is we're going to stop here, and then we're going to do our next video, this kind of last part of our filtration series here, looking at the extrinsic controls. And it'll be a relatively short part, but we'll look at those that are occurring outside of the kidney.